Hi, I'm Professor Steve Forsythe, and this video I'm going to pick up the story about a 10-year analysis of a neonatal intensive care unit outbreak that was caused by Cronobacter sakazakii. Now, this is the second part. The first part was focused on the pulse field gel electrophoresis and background to Cronobacter issues. There are a couple of other videos I've already done on YouTube, one about strains which were isolated from Michigan by the FDA, and another one looking at Cronobacter sakazakii pathovar CC4, which is what I'll be introducing in this video. The main points are already in a publication by myself, Current Trends in Microbiology. But the thing is that there isn't the rationale of why you do something in research that always comes out in a scientific paper. So in the videos, I'm able to add little insights as to what really occurred as such. So part one, they say, that covered sources, misidentification, and what's a clonal organism. I'm going to be picking up on that again in a moment. Then I introduced the actual outbreak, which occurred in 1994. But fortunately, one of the major investigators, Marie-Francois, had the foresight to put those strains into long-term storage for a time when they could be analysed in more detail. And we're really, really grateful that she did it. And we got as far in the last video talking about pulse field gel electrophoresis results. So part two is going to pick up with regard to multi-locus sequence typing. We're going to be looking at CRISPR-Cas arrays, SNP analysis, even capsule profiling, the research that's come out of this one outbreak, as well as key references and who am I? So let's just orientate ourselves. Cronobacter is the name of a bacterial genus within the Enterobacteriaceae. The genus is composed of 7 species, of which one is Cronobacter sakazakii. Now, the previous name for the genus was Enterobacter sakazakii, and some people think Enterobacter sakazakii means Cronobacter sakazakii. That is not true. Enterobacter sakazakii covered the whole genus, of which one species out of the 7 is sakazakii. You can see in the Enterobacteriaceae we've got there Citrobacter and the Enterobacter, but we've also got a couple of other genera which people are not so familiar with, the Franconibacter and the Cicibacter, and they became apparent because of all the research, focus research by other groups on the Cronobacter, and it revealed a greater diversity within the Enterobacteriaceae. I mentioned in the last video about a clonal organism. Again, this is something which is often overlooked. We pick up differences in DNA because of mutation, transformation, transduction, but if those mechanisms don't occur, if those mechanisms of change in DNA are corrected, and there's a restriction on the DNA that's inserted, then you get a clonal organism, which is essentially what you would get 1, 2, 4, 8 divisions of microbes and so on. And this occurs with Cronobacter sakazakii. It also occurs with Salmonella enteritidis. It almost certainly does not occur with all Cronobacter. I'm very certain it does not occur with Cronobacter dublinensis, for example. The thing is, you will still get a result. You will still get the same banding pattern on pulse field, but it does not mean those indistinguishable strains are related, because unrelated strains can give you the same banding pattern. So indistinguishable does not mean the same. So as I say, this is going to be looking over publications over a 10-year period starting 2007. We're going to make our way over here to 2017 via various papers. And as in the last video, we are talking about this outbreak in France. Marie-Francois, the person who collected the strains and put them into long-term storage. And these strains in this paper were then known as Enterobacter sakazakii, but for continuity purposes, I've already told you this was the one species called Cronobacter sakazakii. And they came from a range of babies, and we also have isolates from prepared feeds and end of bottle as well. When we did the pulse field gel electrophoresis, which is what you did in those days, then we found that the strains divided into 4 groups, and 3 babies died who had the pulse type 2 strain. There was a 4th baby that died, sadly, and when we came to analyze using 16S, it turned out that was a misidentified Enterobacter cloacae and was not sakazakii. The summary of that was pointing out that misidentifications do occur using phenotyping, pointed out that the pulse field did divide all these strains of Cronobacter sakazakii into 4 groups, and that now we find out we didn't have one outbreak by one strain, we actually had several overlapping outbreaks by different strains. We even found that babies can be infected by more than one strain of Cronobacter sakazakii. 2 pulse types were found in prepared feed and end of bottle, and one particular pulse type, that pulse type 2, was associated with 3 deaths. And I popped in into that paper briefly that maybe there's a hint that some Cronobacter sakazakii are more pathogenic than others. And what I pointed out was, because everybody focuses on the powdered infant formula, but you need growth. And these formulas were prepared, they were rehydrated, put in a big syringe, and slowly those syringe contents were pumped into the baby. They weren't individual feeds. So ample opportunity at room temperature for contaminating bacteria to grow, also to attach to the tubes, make a biofilm. Please see previous video for more details. Now, big jump. In between that 2007 and this paper 2009, we had the taxonomic revision that Enterobacter sakazakii was a unique genus called Cronobacter, and what we started to do was to divide that down further using multi-locus sequence typing, because 16S was clearly not sufficient. It didn't differentiate strains well enough. There are various limitations on 16S with the organism. So this is a key paper on multi-locus sequence typing, the initial one, 2019, and there is a YouTube video specifically on this. So I'm going to move on. The MLST database for Cronobacter is this one, pub-MLST. You can submit whole genome sequencing or the old style, 7 loci. If you are doing 7 loci, bear in mind that there are 2 suggested primer sets now, because some people sometimes have trouble with GlinS. The database, as of a couple of days ago, there were over 3,000, almost 3,500 isolates in that database, and there's over 1,000 genomes, whole genomes in there as well. And I'm the curator for all this. So, multi-locus sequence typing. When we make a phylogenetic tree, we get a structure. We've got this 7 species going down here. And I was pondering this, and it was, in fact, it was a Saturday morning. I was just there. I was trying to work out, is there a pattern? Do some species come from particular countries? Do some species come from particular sources like plants or animals or infant formula or particular countries? And I looked at clinical, and it was a bit fuzzy. I couldn't get a clear pattern. Maybe, that was it, but I wasn't satisfied. And the problem with clinical, when you look up strains which are clinical, is you need to look at the context, because that strain may have come from blood and therefore very significant, or actually it may have come from, in this case, a baby's poo, and maybe the baby was asymptomatic. That is still a clinical isolate. But what's the significance of that? So what I did instead, I put that to one side, and instead I only looked up the strains which were from normally sterile sites. So I looked at strains from cerebral spinal fluid, and bang, that's when the pattern came out, because for bacterium to be in the cerebral spinal fluid is highly significant. From that starting point, we then came to this paper of 2011, looking at or announcing the predominance of Cronobacter sakazakii sequence type 4 in neonatal infections. In actual fact, you can just see in the abstract here, in particular with regard to neonatal meningitis. So what that paper, and it's one of my shortest ones, I think it's only about 600 words, and it put out that there are clearly definable pathovars. For neonatal meningitis, many of the strains are in the sequence type 4, or now known as clonal complex 4, but those with necrotizing enterocolitis tended to be sequence type 12, and when we come to adult infections, it's primarily clonal complex 7, Cronobacter melanoticus. So out of the 7 species, the ones which are more clinically important are the sakazakii and the melanoticus. Melanoticus tends to be more an adult infection organism. Sakazakii tends to be more an infant organism, and within that we have ST4 more with meningitis and ST12 more with necrotizing enterocolitis. Now, when you publish a paper, obviously the date of that publication can be like 6 months or thereabouts, more or less, from when you submitted that paper, and obviously the work was done for several months or even years before then. But that paper came out in 2011, Emerging Infectious Disease, the CDC journal. But coincidentally, sadly, another event occurred in 2011 at the end of the year, and that was this. Thursday, December the 22nd, just days before Christmas, 2011, Walmart recalls baby formula after infant death. Please bear in mind, this headline is not based upon epidemiological evidence of strain in powder, strain in baby. This is inference. That's what the article is based on. But that publicity led to a fall of 12%, 11% in the shares of the manufacturer, because the baby was only 10 days old that had died, and sadly you could even see a video of the parents very distressed talking about it on social media. So what we did, you can see where the factory was. It was in Michigan. So what we did, we contacted CDC, and CDC kindly sent us all the strains that they had received for 2011, not just associated with that particular story, but all their strains they had received for 2011. Now, because of the time it takes to write the paper, submit it, etc., or do research as well, this paper came out in January 2013. So December 2011 was where it hit the headlines. January 2013 is when the paper came out. And these were all the strains that CDC were able to send us. And they're from different states, and they are different sequence types. But when you look at those which were associated with meningitis, it again pops out as being the sequence type 4 or clonal complex 4. So our initial work was retrospective. It was looking at strains from decades before. Now, the same year, 2011, we have these other strains: cerebral spinal fluid, meningitis, current isolates, current concern, and they're sequence type 4. So that roughly made sequence type 4 almost predictive of the meningitic potential of that variety. Now, one of the things which we've also done is we've worked with previous authors of previous papers, and we have re-identified the strains. And what we did, one paper, is we contacted authors of well-known papers before. This one's from Germany, strains from Germany from factories and infant powders. This one's from various factories in Australia. This one's an international study from Harry Mountains way back in 1988 for international strains. And we re-identified all those strains, and we did the sequence type. And of those infant formula strains and those from factories, 25% of them with a meningitic variety. So it's highly prevalent in infant formula. And those are studies which are like independent of each other over a very, very long period of time as well. If we take the MLST and look at the evolutionary history, how the DNA may have recombined, etc., then we get this like funny cobweb picture. We have Cronobacter sakazakii here clustering. Here we have Melanaticus clustering, Mauchensky and Dublinensis, very, very diverse species indeed. Bear in mind that by 16S, you cannot reliably differentiate Sakazaki from Melanaticus, so you're way beyond the differential potential of 16S here, way beyond. You're another universe almost. So put 16S, put it to one side. MLST really divided it down, and the clonal complex is where ST is distinguished by less than 3. If we focus in, what we find are that strains within the same clonal complex are clustering together, which is what we predict. But that's only 7 loci. What if we take those same strains and instead we do it by ribosomal MLST, which is looking at 53 loci in the ribosome assembly complex, and we get the same clustering. So strains which were clustered based on 7 loci for classic MLST, they cluster again when you do utterly different 53 loci. So here's CC4, here's ST8, here's CC7. If we then expand it to core genome MLST, this is a default one in the pubMLST database of almost 2,000 loci. Again, the same strains cluster together, and this is reflecting the clonality, but it's also reflecting that you're using conserved genes. And that's something which I want to emphasize here, the difference between core genome MLST and whole genome MLST. Much of the work which I've done, published, is often core genome MLST, but we must remember there's far more than the core. The core, if we got 2 strains of Cronobacter here, you got like similar genes, a little bit of variety. They're the core genes. They're the glucose metabolism and protein synthesis. They can't alter too much because maybe they'll lose function and then the strain will die. So it'll vary a little bit, but not too much. Then we have the variable genes, and these are the ones which vary, maybe different plasmids in some strains, different antibiotic resistance factors, CRISPR-Cas arrays as well. So these are the variable genes. They are what are also included in the whole genome MLST. So core genome and whole genome MLST are not the same. And just to emphasize this, is a very handy table comparing core genome MLST with whole genome MLST of Salmonella. And for the Salmonella, this is a pan-genome. There's a huge number of strains they did for this. The core genome, 3,000 loci. With regard to the whole genome, it's 21,000. So core genome is the core, it's not the whole. So that leads us to moving away from the core and looking at things that vary. And what we moved to was looking at the CRISPR-Cas arrays. And this work was coming up to date now, 2016, 2017. Now, an easy way of just explaining what's the CRISPR-Cas arrays is to think about human differentiating identical twins. Identical twins have by definition, a high degree of clonality. And if we pretend for a moment, just suspend reality, but if we just pretend for a moment that these clonal twins can give birth to clonal twins, then in time, we're going to have a lot of clonal twins. And when you look at them, you're not going to be able to differentiate between them. And if you're a Time Lord and you got into TARDIS, and those people in the UK will know what I'm talking about, you jump in your TARDIS and you pop out of your TARDIS at different times, different years, you will see the same people there. So how are we going to differentiate this person from that person? They look the same. Well, they may differ in their history of being infected, whether by COVID or flu or measles or something like this. So this person will have antibodies against COVID, whereas this twin does not have those antibodies. This person may have antibodies to flu, this person doesn't. So you differentiate not on the core, but on the additional immunity. And in bacteria, that's your CRISPR-Cas arrays. So we have a leader sequence. We're going to look at those very, very briefly. And then you have this, and it's like a tape recording in a sense of which phage and plasmids the strain has been exposed to. Essentially, this tape recording is based upon a fragment of DNA of the foreign DNA, and it's made so it will act as an antidote if it gets infected a second time by nearly the same strain. That is very, very generalized indeed, but please bear with me. So if we say, okay, here we've got a strain, and it starts here in America, goes to Africa, goes to Russia, goes to Australia, goes to South America, goes to Europe, it's like that strain be exposed to different phage in the sequence reflected here. So we have a timeline. If we have a strain and it started in America, and as it divided, it went to Africa, but it did not go to Russia. Instead, it went to South America, then up to Europe. Then it's got a different timeline. So we'll be able to differentiate those two strains based upon the phage it's been exposed to. In Cronobacter, the leader sequence, there are two types of leader sequence for those who want to know, 1E and 1F. We're going to focus on Cronobacter sakazakii CC4, and for those who want to know, it's subtype 1E. Put that to one side. We want to look at that tape recorder, and there are different recordings made. When you look at the first array, they're all the same. Absolutely no differentiation of this memory tape, even though those strains come from different decades and different continents. So we look at the second site of recording of the timeline, and we've got these strains, and here's the French outbreak ones, and there's like only one memory difference. Here are other unrelated strains, really, and they've got two differences. So the CRISPR-Cas memory has almost like fixated. It hasn't continued evolving, recording what the strains have been exposed to. It's absolutely remarkable. We really expected variation here, but this is the clonality. It is so, you got strains from different continents, different decades, and the CRISPR-Cas arrays are extremely similar. And you can see these strains, they're from your French outbreak, and they're indistinguishable from each other. So let's try SNPs. And I'm going a little bit backwards. This was like 2 years before we looked at SNP analysis of these strains. The original paper, we did pulse field gel electrophoresis. When you do SNP analysis of those 4 pulse types, yeah, total agreement. SNPs divides them into 4 groups. The pulse type 2, which is the ones where 3 babies died, those are now known as the ST4s. These pulse type 1 are ST12s. We got clonal complex 1 here. These were the formulae that were fed to the babies, were prepared. And then we got pulse type 3 here, which was ST3. If we just focus on the ST12 strains, it's these strains here. Look at this phylogeny. They're the strains where 2 of the babies had necrotizing enterocolitis, some were asymptomatic. We don't have any deaths. The first baby, we don't have details, but that particular outbreak has gone from the 3rd month through to June. So we've gone from March to June. And when we do the SNP analysis, we can see we got 3 strains which are similar up here and 3 here. And the SNPs differences are quite large, quite significant. Excuse me. So let's move on. These are the cluster 3 strains, the ST13, and one's from baby, the others are from end of bottle, and also from prepared feed. So they're extremely similar. Very, very few SNP differences amongst them. Then we've got these, pulse type 2. These strains were roughly indistinguishable by pulse field. You again, the show of pulse field, I mean, you get some strains which give no results whatsoever, and then certain like experimental variation you get. So indistinguishables in like inverted commas there. All those strains are ST4s by 7 loci. In actual fact, they're indistinguishable when you do 1,800 odd loci. Again, they're still indistinguishable. And they all had the same CRISPR-Cas timeline of being exposed to phage. So they're incredibly similar to each other. So they're the ones here where we had 3 deaths. We got our meningitis here, we've got necrotized enterocolitis, we've even got ESBLs. And we got baby B here. And 3 of these isolates are from the same baby at different times. And baby B also had an earlier infection with a different strain. So poor baby B. When we do the SNP analysis and detailed analysis on top, we find that the majority of strains cluster together, including the prepared feed. Very, very, very similar, few SNPs. But baby B, the 3 strains of pulse type 2, 3 strains of sequence type 4 from baby B are 300 nucleotide differences. And the reason being that the strains from baby B lacked a phage insert. Absolutely amazing. Not pulled out by pulse field gel electrophoresis, not pulled out by conventional 7 loci MLST or core genome MLST, not pulled out by CRISPR-Cas arrays, but by going to the nucleotide level of the whole genome. It turned out these 3 strains lacked an actual bacteriophage. Remarkable work. Now, what's the speciality? And one has to be careful. We haven't got time really to go into clonality effect on looking for virulence factors and the problems of co-inheritance. But if you're interested, then please look at this paper on the capsule profile. Again, 2017, so it's like part of the 10 years. Because what we found was that strains associated with meningitis, primarily the ST4s, but because of co-inheritance, it was significant. We found other meningitic strains, they had the same capsule profile. What we find is that this is a capsule profile for strains associated with meningitis. That's a capsule profile you find with your CC4 strains and some other strains which also are associated with meningitis. So please have a look in this more detail. These are agar plates. This is a strain on milk agar, rather appropriate one, milk agar. This strain is not capsulated. This is one of your CC4s on milk agar, and the agar plate is actually upside down on a scanner, flatbed scanner, obviously covering on it, and so it's upside down plate, and you see the organisms dripping onto the lid. That's how much capsule, and so you have changes in expression of capsule, but the capsule composition is the key thing. And the genes for K2, the K antigen, the region 2 is very different to the region 2 of the majority of strains. Majority of strains have K1 antigen. CA2 is a colonic acid, and cell is the production of cellulose. So where are we? 2007, published a paper on a 1994 outbreak. In total, we have published at least 30 papers where the core findings were based upon these strains from this outbreak. There was an associated, we were sent later, another strain that was found, I think, a boy's leg infection. They said, oh, do you also want this strain? Yeah. It's unrelated to the outbreak, but it occurred similar unit, and that led us to the new species of Cronobacter universalis. It also led us to finding that sometimes the potential of pathogenicity, the virulence, may be due to outer membrane vesicles. It led us to recognizing the issue of clonality and the issue over Cronobacter pathovars. And we've gone all the way through from pulse field gel electrophoresis, multi-locus sequence typing, CRISPR-Cas arrays, SNP analysis, capsule profile, exhausting list. Bottom line, indistinguishable does not necessarily equal the same. So it's been quite a roller coaster experience analyzing these strains. Fascinating what has been pulled out from them, and a real eye-opener. And thank you to everybody who's contributed to the studies. Some of the key papers, and this is obviously only some of the key papers I've listed here. Most of these links will be active. If not, go to this site where you can download most of them, or if you have problems, just email me. Very happy for them to correspond or assist in whatever way. So who am I? Well, that's me, so-called retired, ignore it. Doesn't really happen. External advisor to regulatory authorities, guest lecturer, and essentially busy person at times exploring the world of Cronobacter and related organisms. So thanks very much for your attention. Please let me know if there are any topics you'd like me to cover in the future, and all the best.